Hello dear students, in the previous lecture we discussed the step number 7 of glycolysis, in which we ended up with the formation of 3-phosphoglycerate along with the release of ATP. Now we are going to talk about the conversion of this 3-phosphoglycerate into 2-phosphoglycerate. The reaction takes place by the help of enzyme phosphoglycerate mutase in the presence of magnesium ions. So you might remember that mutases are the isomerase enzymes that catalyze the shifting of a functional group from one position to another within the same molecule. And here we are going to talk about the shifting of phosphoryl group from position number 3 to position number 2 of same molecule. So actually the reaction takes place in two steps. So as the first step, what happens is that the histidine residue which is present at the active site of the enzyme, it gets phosphorylated itself by the addition of a phosphoryl group, and now this phosphorylated enzyme reacts with the hydroxyl group of carbon number 2 and transfer its phosphoryl group to this position, and in this way it forms an intermediate which is known as 2,3-bisphosphoglycerate. So this 2,3-bisphosphoglycerate is actually the intermediate which can also affect the affinity of hemoglobin for oxygen in the erythrocytes, and here during glycolysis it is present in very small amount because of the fact that once it is formed it reacts with the enzyme, and the same histidine residue of the enzyme is again phosphorylated by 2,3-bisphosphoglycerate by the transfer of phosphoryl group attached to carbon number 3. In this way, the enzyme is again converted into the phosphoenzyme, and this intermediate is now left with only one phosphoryl group that is present at position number 2. And during this reaction, the histidine residue acts as an acid catalyst because it replaces the phosphoryl group with its hydrogen ion, meaning it donates the hydrogen ion for the formation of 2-phosphoglycerate. But the point is that what could be the possible benefit of this step? So when we look at the molecule of 3-phosphoglycerate, we can see that there are two negative charges, but they are found at quite a distance. And when this 3-phosphoglycerate is converted into 2-phosphoglycerate, the negative charges of carboxylate group and phosphoryl group are brought together, and this means that this molecule is more reactive and will be slightly higher in energy, and therefore it will be easier to transfer the phosphoryl group from this molecule. So now we have got 2-phosphoglycerate and we want to generate a molecule with a higher phosphoryl transfer potential. Therefore, there is a dehydration reaction carried out by the enolase enzyme in which water is lost and there is a formation of double bond between carbon number 2 and 3, and phosphoenol pyruvate is formed. So phosphoenol pyruvate has much higher phosphoryl transfer potential as compared to 2-phosphoglycerate. Therefore, it is more likely to transfer this phosphoryl group to adenosine diphosphate so that it can generate adenosine triphosphate. Now with the formation of phosphoenol pyruvate, we reach the last step of glycolysis, and we know that phosphoenol pyruvate is an enol moiety which tends to convert into its more stable form, that is the keto form, because the ketones are much more stable as compared to the enols. So the phosphoryl group is transferred to ADP, and this results in the formation of pyruvate and ATP. The reaction is carried out by the enzyme pyruvate kinase in the presence of potassium and magnesium ions. In fact, this is also a substrate level phosphorylation, and in the beginning, the phosphate group at carbon number 2 is replaced by hydrogen. But this enol form of pyruvate is very unstable and it tautomerizes rapidly to its keto form. The overall reaction has got large amount of negative standard free energy, and about half of the energy which is released by the hydrolysis of PEP, that is around 61.9 kilojoule per mole. This is conserved in the formation of phosphoanhydride bond of ATP, and remaining is actually utilized to push the reaction towards the synthesis of ATP. So importantly, pyruvate kinase reaction is essentially irreversible under the intracellular conditions, and this means that the net amount of energy which is produced from the breakdown of one molecule of glucose is 2 molecules of ATP during glycolysis, which takes place under the anaerobic conditions. So this brings us to the end of glycolysis, and with this the present lecture comes to an end.